So maybe I like to start our session. As I show you in the opening welcome remark, I think our effort should be focused on how to deliver safe and effective stem cell therapy to many patients at affordable cost. And our strategy is probably similar to same as others, is multidisciplinary collaboration, multi-sector collaboration, and international collaboration. We often called open innovation. And at this session, we'll focus on multidisciplinary and multi-sector collaboration. So key target is, I think, large-scale production of high-quality stem cells, such as pluripotent stem cells, and also differentiated cells for actual transplantation therapy. And all steps and procedure eventually must be at lower cost and high-quality control for practical use. And I think particularly the key technology is now chemical compound, develop chemical compound to replace cytokines and other biological macromolecules, and functional materials to replace biological ECM and others compound. And large scale is a keyword, and quality control is another keyword. So multidisciplinary work, for example, if we can control the stem cells growth and differentiation by small molecule chemical compound or functional material like great use of nanotechnology and others, we can increase the reliability and quality control, and at the same time decrease the cost tremendously. And as one example, I'd like to show briefly our recent work on cardiac differentiation from pluripotent stem cells. So conventional method usually use proteins and growth factors. It's cause high cost and not good quality control. However, new method we developed and probably will be developed in many other purposes is to find out chemical compound which can replace cytokines for differentiation control. And that realize defined and xeno-free and lower cost and high quality control method for practical use. And so we published very recently one paper in Cell Reports. It's actually realized serum-free, cytokine-free, xeno-free cardiac differentiation, and almost protein-free. We still use serum albumin, but almost all protein-free method. It's ideal for clinical usage and other applications which require a high level of quality control. And at the same time, our simple calculation can decrease the cost of this differentiation protocol by 1/20th. And the focus of this event is on our multi-sector collaboration called Open Innovation by academia industry collaboration. I am now leading a government-sponsored project involving these prominent academic scientists and also 8 Japanese companies which are known for high technology and reliable technology in their specific field. So you will hear by several members of this collaboration following the tissue engineering talk by Professor Tabata of Kyoto University. And the central focus of our project is development of automated large-scale culture equipment and quality control system for human pluripotent stem cells lines. And so far, we already developed with a company of such a machine prototype, which use culture bags, gas-permeable bags, to culture the large-scale stem cells, and with a tube and pumping, it can realize automated cell medium change, subculturing, and harvesting in closed system. So it's the best for quality control. And myself is now developing a new three-dimensional culture system of human pluripotent stem cells. And compared to the conventional adherent culture system, it can realize large-scale production and without need of any adhesion protein, for example. So simple is beautiful is our keyword. So that's my brief introduction, and I'll hand over to the chairperson. Thank you very much.
Thank you for Professor Nakatsuji. My name is Takashi Asai from Kyoto University iCeMS. I'm a moderator of this session. Next speaker is Professor Yasuhiko Tabata, Professor and Chairman, Department of Biomaterials, Institute for Frontier Medical Science, Kyoto University. I just introduced his career. Dr. Yasuhiko Tabata is a professor and chairman of Kyoto University, and also he's a professor of the Graduate School of Medicine, Osaka University, and also he's several other universities visiting lecturers. And he has published over 1,000 science papers, including 120 book chapters and reviews, and he has 140 patents. And also, he received many awards, including international and also Japan award. Dr. Tabata is a board governor of the Tissue Engineering and Regenerative Medicine Society International, and also there are lots of Japanese society he joined. His research is very interdisciplinary in nature and bring together the field of polymer chemistry, pharmaceutical science, biology, and basic and clinical medicines. So, please, Professor Tabata.
So thank you very much. Too kind introduction. It's how long can I talk? How long? 20 minutes? Okay. So thank you very much. Before the presentation starts, what I would like to appreciate is my colleague, and thank you very much. Give me a good chance to come here to then share the time. So what I'm talking about is some biomaterial things. I'm not a stem cell people, but is using the stem cell to realize of tissue regeneration of the based on that regeneration therapy, and also that is a stem cell therapy, stem cell research. So then the biomaterial is very, very needed. So what is the biomaterial thing? So then maybe that audience here, the biomaterial equal to a medical device. So but I don't think that is some biomaterial not limited to the medical device. So biomaterial is a meaning of that materials is using in the body, or that contacting of that with the cells, or that the cells, or that the proteins. So that meaning, so that the biomaterial is very, very important to realize of the regeneration therapy and also that stem cell biology. So today I'm focusing on that, just how to use a biomaterial to induce your own healing potential. So then finally we got is a sum, is a tissue regeneration, is a translation research. So today is due to the limited time, so that in my chair, so that is said to me, so just only 20 minutes. So 20 minutes is not enough to talking the everything. So just focusing of that is how to use of the biomaterial to use tissue regeneration. So today is my talk about that is some biomaterials, technology of the indispensable the regeneration therapy and also that stem cell research. So what is the regeneration therapy? So I think that is every person have the different definition. So but I think regenerative medicine is induce your own healing potential. Finally, we got some regeneration therapy. So that is some healing potential based on that is some stem cell is a potential of the differentiation of the proliferations. So then is to induce of that is the induce of your own healing potential. So there are the two ways. So one is that stem cell is the high potential of that proliferation differentiation and the proliferate of the in vitro to put it back. The people to expect of that some cells is activated to induce their own healing potential. Finally, we got is some regeneration therapy. So that I think another way, so that is another way, so that which is I'm talking about today. So when you use of that the biomaterials to proper way to induce or healing potential, the final we got is some achievable tissue regeneration without any cell transplantation from the outside. That's focusing on today. So here I'll show you that is structure of the body tissues. So there are the two components of that constituting the body tissues. So one is the cells, another one is the cell environment. So I think I would like to explain that is what is the environment is if that is some cell compared to my body. So without any food, without any house, my body is weakened. So that situation is similar to the cells. Even if that cell is a very, very potential, without is a house, without is some food, cell is even. So that is some weakened. So then maybe that what is the house of the cells? So that is some extracellular matrix. So what is the food of the cells? So that cell growth factors. So then you have to use using the proper way the biomaterials to use of that growth factors, and that is some scaffolding system. So when you apply to your body without any cell transplantation, so then the cells are activated in the body to induce of that some tissue regenerations. So here I'll show you that is four methods to induce of your own healing potential. Finally, we got the regenerative medicine. So the first one is that we prepare that is some biodegradable scaffolding system to apply that. Another one is that with the cells of the scaffolding, with a growth factor with the scaffolding. So both of them, so that is some scaffolding is a combination of the cells and the growth factors. Put it back to defect. We are waiting for that is some tissue regeneration after the defect. So just due to the limit of time, now focusing on that the growth factors. If that we have the growth factors, so growth factors might be the enhance of that the proliferation of the cells in the body. So, but is when we have the growth factor in the solution injected into the body, we cannot always expect of that biological activity in the body, in the growth factor, because due to that is some in vivo instability. To break through that problem, we need some technology. That's one is a drug delivery system. So using this kind of the slide, so then I'll show you that what is a drug delivery system. The point is drug delivery system is the drug will be combined to the biomaterials to induce or that enhance of the biological activity of the drug. So here there is four purposes of the drug delivery system. The first one is the controlled release of the drug. The second one is the prolongation of the drug lifetime. And also third one is acceleration of drug absorption. Is a fourth or the drug targeting. Due to the limit of time, I think the full purpose of that is the drug delivery system is applicable for induce your own healing potential. So what is the drug for that tissue regeneration therapy? So that is the growth factors. So here one is that to make that it's a story short, is a growth factor. I'm interested in the use of that the growth factor, basic growth factors. That one is one of that angiogenetic factors. When I have that it's a basic FGF in the solution, so inject on the back of the mice. So then this is the tissue appearance 1 week after the injection. So unfortunately, we could not observe that is angiogenesis here because that the basic FGF injected immediately after that, so injected excreted from the injection site. So but breakthrough of that problem, so that I said that you have to use in a proper way a drug delivery system. So that is hydrogel will combine to that basic FGF injected with it. So without saying anything, we could observe that is angiogenesis here. When you have that some growth factor, which have that is a food of that is any type of the cells, so that a combination of that growth factor is a release system, we have already succeeded over the control release of the growth factors. So, and also that here is that if you will take that as some hydrogel release system, we could observe so that the some succeeded in the regeneration therapy. So here, so I'll show you that. Oh, I'm sorry. So here we prepare that ischemic heart model. When injected the basic FGF solution, we could not observe the angiogenesis here because that after the injection of the basic FGF disappear from the injection site, as I said. To prevent of that is the kind of the problem. We prepare that some small particles incorporated basic FGF injected intramuscularly, we could observe that angiogenesis here. So, and also that when injected after that, we could observe that is heart muscle motion recovery. And also that when we prepare that small particle incorporated another type of the growth factors, inject of that is some bone tissues defect, so we could observe that bone regeneration. And another type that we prepare that small particles, incorporate of the base, incorporate of that is a growth factor, which is the food of that is some cartilage cells. We inject of that the cartilage cavity, so we could observe, so that is some regeneration of that cartilage, so which might be effect of that is some osteoarthritis. Okay, here I have already introduced of that the growth factor system induce your own healing potential. I mean that is some especially the angiogenesis. So there is a two purpose of the angiogenesis. Why induce of the angiogenesis? First one is that is some ischemic disease therapy. We inject of the small particle, incorporate angiogenesis locally. That is the first one. So another one, so that in advanced angiogenesis for the cell transplantation, to apply to the angiogenesis to supply to the nutrition of the cells for that. So I'll show that. So then here, I prepare that ischemic leg model. So then we prepare that is some small particles incorporated base shape, which is angiogenic factors for the control release locally. So when injected of that, so we could observe that angiogenesis. But as a control, when injected a base capture solution, we could not observe that angiogenesis here. So based on that, we have already got is a feasibility of the using of that animal study of that monkey model. So then based on that, we are translated to that is some human trials. So here, so that here, what is that? Oh, you don't have to move it because that is some, skip the next slide. You know, this guy is some 27 years old suffering Berger disease. So this guy, you know that's how many toes so that you have that 1 foot. So, I'm sorry. So then 2 fingers, 2 fingers have already amputated. So this guy went to that university hospital, received of that cell transplantation therapy. So, but it's unfortunately that failed. When it come to the Kyoto University, because that we have already got is some kind of that biomaterial base of that tissue regeneration therapy approved. So then, so after the injection of that, so that 4 weeks later, getting better, the 12 weeks later have already done. You know, this kind of an approach, when you will take the hydrogel in the proper way, induce your own healing potential. So we got is some achievement of that realization of that regenerative medicine without cell transplantations. So then we have already done this kind of the clinical experiment. So then is some 35, actually that is have already done 40 patients. So then it have already. So that's again, so that is a biomaterials, it's a use of that proper way to induce some healing potential. Finally, this is not science fiction. Because what is the final goal of that regenerative therapy? So that is not science fiction, not writing a paper to treat a patient. So that I'm talking about. So that is how to apply to that is developed in the university to treat a patient. So another case, I'm sorry, because that you have lunch very bloody. So but this is again, so that is a very, very severe case. When applied to that is some growth factor release system, is put on that is some collagen sponge with that is a scaffolding, so which is that is house of the cells, we could observe that. To tell the truth, we have already treated this kind of that is some 7 patients. Out of the same patient, 4 patients are already cured. 3 patients not cured. But when I asking, when talking about that this kind, so I'm always asking about that, why that 3 patients not treated, not healed. So, but I don't understand because that I cannot say because that is in Japan. If this is the case, so that the cut of that amputated of foot, so that burning it, so we can't do that. But is when we brought of that this kind of that technology to another Asian countries, so Asian country of that they have the different rule from that is Japan. So then they have already done so that even if that fails, so that foot is amputated foot, so then it's examined. So they said that is what happened in the human is that is similar to the animals. So that's meaning, so I have that as a very intent of the feeling, so that is a proceed of this kind of experiment. So another thing that is less severe. So when applied to that is some hydrogel system in the alveolar bone, we could observe that alveolar tissues. So here is a very looks better, too easy to understand for here. So this is a cement line here. When applied to the base capture solution, basically activated of that cells implanted over here, presented here. So then is that some bone is elevated of that is some 8 centimeters. again, so that you have to take over the biomaterial the proper way to induce your own healing potential. That is a biomaterial based on that tissue regeneration. Okay, so then without any cell transplantation, so as I said, that is some hydrogel system for the examples of the control release of the growth factors. So we have already done, so that is 308 patients have already healed. Just only that is some biomaterial technology. So I cannot deny the possibility of that, the importance of that cells. But what kind of the cells from the viewpoint of that some patient, because that is adult stem cell is very close to, because that is an institute of that discovered the iPS cells. So, but iPS cell is a little bit far from that is clinics. So I want to take out adult stem cells. So here we prepare that is a ischemic model. If that is the infarction site is very large, very small, as I said, that when injected of that small particles incorporate angiogenesis locally, that's going to be fine therapeutically. So, but when its infarction site is very, very large, so we need of that combination of the stem cell transplantation. So, but even if the stem cell transplanted, so that the stem cell is easy to die because of new nutrition, new oxygen supply. So breaks up that problem before that cell transplantation. So we injected those small particles, incorporated the basic FGF, induced angiogenesis locally, followed by that is some cell transplantation. So here is a cross-section of the heart of the monkey. When applied, so this is that is some disease. This is very thin. When applied, that is a cell transplantation. Then looking at here, grafting rate of the cells, 5 to 10%, a little bit thick, but it's not perfect. But when applied to that is some growth factor release system induced of the angiogenesis locally, followed by that is some cell transplantation. So that is very thicker that is some normal area, and also the grafting rate of that cells transplanted, 30 to 83%. And how about that is a cardiac function? So we got is some synergistic effect. So here, so that even if you got is a very, very good is a high quality of the cells, so but is a when injected without any modification, that is not good. You have to combine to that the biomaterial technology. Otherwise, you cannot expect always for that the therapeutic efficacy of the cell transplantation. So here, based on that, we have already started over that the clinical trial. So then we pick up that is some precursor cells from that heart muscle of that patient. So expanded or put it back. Of course, the combination of that the hydrogel release system, we could get is some very good is a result. So here, so that is some growth factors of the cells that are there. So that is just only, as I said, that is when applied to that the growth factor release system. So that is some cells that are present of that is activated. So then is a healing potential. We got is a type of that tissue regenerations. So but is some cells that are not there. So we have to increase of the recruitment. So then to apply to that the growth factor if necessary. So then is you may know. So that is some SDF1, so just one that is some chemokine to induce of that some cell recruitment. We applied to the chemokine in the solution. Unfortunately, we could not observe that there some angiogenesis because the chemokine immediate after injection, so that is a chemokine excluded from the injection site. As I said, that is a similar way to that is a basic FGF case. So breaks of that problem, we prepare that as a hydrogel, BMP SDF1, to apply that, we could observe, so then is the angiogenesis. What is the mechanism there? So that if when applied a local release of the enhancement of that cell recruitment, so then the cell is recruited of that is activated to that we got is some base stem cell is based on that tissue angiogenesis. So as I said, that we have that is some hydrogel system. Hydrogel system, it can not only one type of the growth factor, but only two types of the growth factors. So we prepare that is some hydrogel is a control release of the SC1, that recruitment factors, and also the BMP2. So that one is some osteogenic activity factors. So here, we prepare that is a ischemic bone defect model. When applied to the SC1, we could not observe the angiogenesis. But when applied the BMP2, so that is the osteogenic effect. So then we could observe that bone regeneration. So but when applied first SC1 and followed by that BMP2, so synergistic effect. And when looking at that is some osteogenic gene expression, so only that is a dual release system, we could observe that high efficiency, high expression of that is osteogenesis. So again, so that is some hydrogel biomaterial system, for example, so you have to use it. So but today, so just I'm focusing on that this kind of the hydrogel system applicable for the control release of the growth factors, chemokines. So but it's Dr. Nakatsuji said, so that he discovered that the low molecular weight drug, if you take the low molecular weight or drug, and also that the hydrogel is very applicable for the control release, even that low molecular weight drug. So that is unfortunately today that we don't have to get into because due to the limited time. So another thing, so that is some house of that is a scaffolding system. When applied to the scaffolding, so the resultant scaffolding, the cell is not activated. So I will show you that some. So here the very classical one, so biodegradable polymer the sponge, that is a scaffolding system. When applied to that some bone defect model or the another type, this is a human trial. When applied to that is some hydrogel, the burn, so then we could observe that is a dermis. Dermis of that is some skin dermis regeneration. When applied to that sponge surface, some type of that scaffolding system, so edaphox is the regeneration, trachea, peripheral nerve are the dry matters. So, but is some collagen is a people to use very often to use for that scaffolding because that is some cell is like to the high collagen. So, but collagen is a many type of that problem. One of that big problem is the collagen sponge is very, very weak. So, prevent of that to reinforcement. So then prevent of that problem is the weakness of that is some mechanical weakness of that collagen sponge. So we incorporate of that is some fibers of that is that is a ceramics to reinforcement. Here, so that collagen sponge incubated, so that cultivated with the cells, that's shrinkage. But after that is some collagen incorporate of that is some fibers of that is some granules to reinforcement. So even after the culture, so that is some not shrinkage. So that is again, so then is this kind of that mechanical reinforcement is applicable for that some very works of that the in vivo, so that stem cell is a scaffolding system, and also, so today the little limited time that I can't get into, so but this kind of reinforcement of the scaffolding system also that the cell is substrate for that stem cells in vitro experiment. So when I go to the Egypt, so I have that collaboration with Egyptian people. So then it's he picked up that this kind of one. So then this is a clinical case. So they isolated of the stem cell from that periodontal membrane. So put it back. So then here. So before that treatment, this kind of that is some gingiva is not good. When applied to this one, so we could observe so the regeneration of the gingiva tissues. When you take of that collagen without any reinforcement, so that is not happen. So that's again. So that is some. Biomaterials technology modification is very, very important if you want to use of that the stem cell transplantation, of the stem cell therapy, so that to expect of that is some therapeutic efficacy of that cell transplantations. So next one, if you will take of that is some hydrogel for the control release system, the plus combined of that some hydrogel sponge of that some scaffolding system. So what I have done. So we have a lot of things. So this is very, very great things. So we prepared that is a regeneration of that broncho cartridge. What I have done, so that we prepare that is some tube-like scaffolding system. So isolated from that human, that stem cells, so put it back to expand it. So then combine to that growth factor. In this case, TGF-beta1, because TGF-beta1 is very, very effective to accelerate of that is some increase of that cells which have that potential differentiate into that some cartridge cells. When it put it back, so we could observe that is this kind of that bronch cartridge regeneration. So two things, the very, very interesting things. One is that the very large one. Large is size is bigger than the humans. And also just when applied to that combination of that the growth factors of that cells and the tube-like sponge, so that we could observe so that the cartridge and also that the cartridge, is histology, its integrity is perfect. So again, what I'm talking about. So yes, okay, I have to stop it. What I'm talking about. So that the biomaterial is very, very important. So but you can use so that is in a proper way to induce the immune healing potential. Finally, we got the regeneration of that therapy without any cell transplantation. Okay, so we have to finish it. So we have to go back to move to the end one. This is kind of very, very important things because, I have many collaborators working that is some different fields, and also that's the working of that is some people that are coming from the abroad, and also that private company. Without a private company, we cannot make a dream. So because that is without these commercialization things, we cannot treat a patient. because that is some tissue regeneration based of the regeneration therapy. That is not writing a paper, it's the final goal. So treat a patient. Thank you very much again. Thank you.
Thank you for Professor Tabata. Next speaker is Dr. Wado Akamatsu, Assistant Professor, Department of Physiology, School of Medicine, Keio University. He graduated for Keio University Graduate School of Medicine, and he got his PhD, same section. And also, he went to the University of Toronto, Canada, then go back to as an assistant professor. He's now in the Hideyuki Okano, Professor Hideyuki Okano's group. Please talk about that.
Thank you for kind introduction, Dr. Sada. So today I will talk about the neural stem cells induced from pluripotent stem cells or somatic cells. So currently, iPS cell studies in neuroscience are mainly classified for two categories. Okay. One is disease-specific iPS cells, and the model for the various diseases to analyze mechanism and to perform the drug screening. Another one is the future cell therapy for patients. So iPS cell-derived cells expected for future autograft transplantation. So in both studies, it is important how to obtain neural stem cells robustly and quickly. So especially we are focusing on the spinal cord injury model by using animal like mouse and rat or marmoset. So neural stem cells are self-renewing and multipotent. To expand neural stem cells in vitro, we use a neurosphere assay. So neural stem cells makes neurospheres by culturing in basic FGF or EGF. So neurospheres can be dissociated into single cells and expanded by recurrent passages. So this method was established in 1992 by Dr. Samuel Weiss in Canada. So we have been studying whether these neural stem cells are effective in the treatment of the spinal cord injury model in mouse and rat and marmoset. So recently, we have shown the effect of iPS cell-derived neural stem cells for spinal cord injury model. In that study, only mature neural stem cells effective for the treatment of spinal cord injury model. So interestingly, these young neural stem cells which produce only neurons did not have any effect for the spinal cord injury models. So recently, we also reported neural stem cells induced from human iPS cells are effective for the treatment of spinal cord injury models. This is our protocol to induce human neural stem cells. First, we dissociated the human iPS cells and make embryoid bodies. So then dissociated embryoid bodies make primary neurospheres. So by using basic FGF. So we can make neurospheres from various iPS clones, and also neurospheres can be passaged by dissociating and also placed in the frozen culture again. So actually, we use tertiary neurospheres for the transplantation because we need to amplify the mature neural stem cells by these neurosphere cultures. But there is a problem. So because it takes at least 3 months to obtain the neural stem cell, tertiary neural spheres from undifferentiated from iPS cells. So it is very long time. So further, in human pluripotent stem cells, the efficiency of neural induction depends on clones. So for example, ES cells KHES1 and 2 established in Kyoto University differentiate into neural cells very well, but KHES3 did not. So to increase the efficiency of the research, we try to establish a short and robust protocol to induce neural stem cells. So I've been in University of Toronto, the laboratory of Dr. Derek van der Kooi. So 12 years ago, he established, he found mouse ES cells quickly differentiate into the primitive neural stem cells by dissociating culture, in the culture, and low-density cultures. So these primitive neural stem cells are leaf-dependent, and they make neural spheres and then turn into definitive neural stem cells, which are FGF2 dependent. So recently, they published a paper in Journal of Neuroscience, so they can make the primitive neural spheres from human ES cells. So we modified their protocol, and we succeeded in making neural spheres from various ES or IPS, human ES or IPS clones. And this is IPS, and ES, and this is these iPS cells from Parkinson's disease. So in all lines, so we have plenty number of neural spheres. So even in the KHES3. So these neural spheres can be differentiated into both neurons and also GFA positive astrocyte. So by using chemical reagents, so actually basic protocol was within 2 weeks. We can obtain neural spheres within 2 weeks, but by using chemical reagents, we can modify the protocol, and eventually we get these differentiated neurons within 10 days, 14 days from undifferentiated neural stem cells. Undifferentiated iPS cells or ES cells, sorry. So the next topic is directly induced neural stem cells from mouse skin fibroblasts. Recently, we have shown the partially reprogrammed fibroblasts can be turned into the neural stem cells. So that system is very simple. So at first, 4 Yamada factors are introduced into the fibroblast, and then these stem cells, these fibroblasts are cultured under the presence of the leaf and the basic FGF. Within 14 days in the floating cultures, so they make neural spheres. So this is a slice of these neural spheres. So they express nestin as a marker of the neural stem cells, and also beta-3 tubulin is a neuronal markers. So this is a neural spheres from ES cells. They're almost similar. But interestingly, the property of the neural stem cells directly induced from mouse fibroblasts are a little bit different from the neural stem cells differentiated from ES cells. So this is a differentiated cells derived from ES cells. So usually young neural stem cells generate only neuron, but very few astrocytes. But from directly differentiated neural spheres, there are lots of GFAP positive astrocytes. So, and also we can see the CNFPS positive oligodendrocyte from directly differentiated neural stem cells. So directly differentiated neural stem cells are very highly gliogenic. So these neural stem cells can be dissociated and they made secondary neural spheres. So in secondary neural spheres, the property of the neural stem cells are highly gliogenic. So this is a quantitative data. So in secondary neural spheres, most of the neural spheres differentiated into astrocytes. So we can these data suggested that these neural stem cells directly differentiated from fibroblasts, differentiated very quickly compared to the neural stem cells derived from ES or iPS cells. So using similar protocols, we have succeeded in making directly differentiated neural stem cells from human fibroblasts. So, within 20 days, so after inducing the 4 Yamanaka factors, so we can see the lots of neural spheres from adult human fibroblasts. So they can be differentiated into basically tubulin positive neurons and GFP positive astrocyte. But the efficiency of the human directly differentiated neural stem cells are a little bit low compared to the mouse stem cells. So actually we are now trying to develop the protocol, and we are trying to test whether the neural stem cells can be used for the autograft transplantation in a marmoset model. So this is the conclusion. The first conclusion is we established a robust and rapid neural induction protocol from human pluripotent stem cells. So just 6 days from iPS cells to neural stem cells, and also 14 days to mature neurons. So the second conclusion is we established a new method to generate neural stem cells from mouse adult skin fibroblasts and also human skin fibroblasts. So these stem cells are very differentiated very quickly, and also this is a model for a purpose. So making neural stem cells by the patient, and also go back these stem cells into the patient. This is thanks to Dr. Hiroyuki Okano, and also these experiments are performed by Dr. Matsumoto, and also the PhD student, Koji Nishihara, and also MD student, Mr. Ando. Thank you very much.
Thank you for Dr. Akamatsu. Next speaker is Dr. Kenji Kawabata, Project Leader, Laboratory of Stem Cell Regulation, National Institute of Biomedical Innovation. Just a brief introduction for Professor Kawabata. He graduated from Faculty of Pharmaceutical Science, Kyoto University. Then he got his PhD, Graduate School of Pharmaceutical Science, Kyoto University. Then he moved to the Division of Cellular and Genetic Therapy Product, National Institute of Health Science. Then he moved to president position, and also he's a project leader. So, Dr. Kawabata, please.
Thank you for introduction. Good afternoon, ladies and gentlemen. My name is Kenji Kawabata from National Institute of Biomedical Innovation, Osaka, Japan. Today, I will talk about differentiation of hepatocyte-like cells from human ES and iPS cells and its application in drug screening. As you know, human ES cells and iPS cells can proliferate infinitely, and also can differentiate into many kinds of cells. Differentiated cells from iPS cells or ES cells are expected to apply to regenerative medicine or drug screening. Especially hepatocytes in the liver are known to metabolize many drugs. So if a technology that can promote the hepatic differentiation from ES cells or iPS cells is developed, then drug screening technology would be accelerated. However, hepatic differentiation efficiency is quite low because there are some steps to mature hepatocytes from human ES cells or iPS cells. So differentiation-related genes are transduced with adenovirus vector in an appropriate step. As shown here, we have reported that 3 genes, SOX17, HEX, and HN4-alpha, was transduced with adenovirus vector into the mesenterm or definitive endoderm or hepatoblast, respectively. And then hepatic differentiation is promoted. However, a further maturation of hepatocyte-like cell is needed for widespread use in drug screening. So we screened the 7 transcription factor genes, FOXA2, SOX17, HEX, HN1-alpha, 1-beta, 4-alpha, 6, all of which are important in the liver development to make a more applicable protocol. There are 3 steps to hepatic differentiation. First step is endodermal differentiation. Then the second step is hepatic specification into the hepatoblasts. And the last step is hepatic maturation to mature hepatocytes. As a first step, definitive endodermal differentiation from human iPS cells are evaluated by FACS or qRT-PCR. When FOXA2 or SOX17 genes are introduced to mesenterm, then the expression level of c-KIT and c-KIT-CR4, both of which are endodermal marker, was increased. So I wanted to find the differences FOXA2 and SOX17. When SOX17 was overexpressed, then the cells was differentiated to not only to definitive endoderm, but also extra embryonic endoderm. On the other hand, when FOXA2 was overexpressed, unidirectional differentiation into the definitive endoderm was observed. These results suggest FOXA2 is the most suitable for selective unidirectional differentiation into the definitive endoderm. As a second step, the definitive endoderm on day 6 was transduced with adenovirus vector, and 3 days later on day 9, hepatic specification was evaluated by the FACS or qRT-PCR. We used the alpha fat protein and CYP3A7 as a hepatoblast marker. When FOXA2 or HN1-alpha gene was introduced to definitive endoderm, alpha fat protein expression level was increased. And also, both FOXA2 and HN1-alpha gene was introduced to definitive endoderm. CYP3A7 level was increased. These results indicate both FOXA2 and HN1-alpha can promote hepatic specification into the hepatoblast from definitive endoderm. As a last step, mature hepatocyte, hepatoblast was introduced, transduced with adenobiospector, and on day 20, mature hepatocyte was evaluated by the FOX or QRT PCR. We used the expression of ASGPR1 and CYP2C19, both which are mature hepatocyte marker. When FOXA2 HN1 alpha or HN4 alpha gene was introduced to hepatoblast, then the expression level of ASGPR1 was increased. And also, the combination of FOXA2 and HN1 alpha or FOXA2 and HN4 alpha gene was introduced to hepatoblast. The expression level of CYP2C19 was also increased. These results indicate that both FOXA2 and HN1 alpha can also promote the hepatic maturation to mature hepatocytes from hepatoblasts. This is our protocol for hepatic differentiation from human iPS cells. We used an adenobiospector system for gene transfer. On day 20, some binuclear cell hepatocytes were observed. This indicates that mature hepatocyte-like cells were differentiated from human iPS cells. And next, we investigated the function of our induced hepatocytes. LDL uptake level were increased after differentiation, and the level was comparable to the primary human hepatocytes. As shown in the right panel, indocyanin green uptake was also increased after differentiation. Then, intracellular indocyanin green was excreted from the cells, probably by the specific transporter. These data shows that functional hepatic cells are induced from human iPS cells. Expression of various kinds of drug metabolizing enzymes was studied in induced hepatocytes. In these figures, a triangle symbol means controlled iPS cells, and red square symbol means our induced hepatocytes, and gray circle symbol means primary human hepatocytes. And the left figure shows the expression of phase 1 enzymes, and the right panel shows the expression of phase 2 enzymes. These figures show that induced hepatocytes could express almost equal amounts of drug metabolizing enzymes to the primary human hepatocytes. However, the expression of some enzymes such as CYP1A2 or 3A5 or UGT1A3 in induced hepatocyte was still lower than that of primary human hepatocyte. This result suggests that improvement of differentiation efficiency is clearly needed. Finally, drug metabolism activities in induced hepatocytes were studied by using 9 drugs. Our induced hepatocytes have the capacity to metabolize these 9 drugs, although the activity levels were lower than that of primary human hepatocytes. In conclusion, our methods employing the transduction of FOXA2 and HNF1 alpha gene represent a useful tool for the efficient generation of metabolically functional hepatocytes from human iPS cells. However, a further maturation of induced hepatic cells is clearly needed in the future. For example, by the 3-dimensional culture or additional gene transfer, and so on. This is the end of my presentation. I am grateful to Dr. Umezawa and Dr. Yamanaka for providing human iPS cells. Thank you very much for your attention.
Thank you very much, Dr. Kawabata. Next speaker is Dr. Chikafumi Yokoyama, Chief Executive Officer, ReproCELL. Dr. Yokoyama, please.
Good afternoon. My name is Chikafumi Yokoyama, CEO of ReproCELL. ReproCELL is a spin-out company from Kyoto Universities, founded in 2003. We have 10 years experience in stem cell business. We have succeeded to develop several cutting-edge products in research reagents and drug discovery field. Today, I'd like to introduce you our several topics in brief. This first slide shows our products of a research reagent covering both on-feeder and feeder-free system. Now we are developing a new types of culture media. It is completely xeno-free and suited for floating sphere culturing. It will be released early next year. This product, primate ESL medium, is the most famous and important product for ReproCELL, which was developed by Professor Norio Nakatsuji for human and monkey ES cells. After releasing that, Professor Shinya Yamanaka used our culture media to establish a human iPS cells. This is a very famous article written by Professor Yamanaka. You can see the very small name in the red circles, ReproCELL. After that, our culture medium became famous. Now we have a top share in Japan in the research reagent field. And this is another type of culture media. Usually, a researcher has to exchange and operate every time, even on Sunday and Saturday. ReproFF2 can allow only 3 days operations, Monday, Wednesday, and Friday. Researcher can take a rest on Sunday and Saturday. It's a new type of culture media. This slide shows our drug discovery products. We are providing 3 types of cells, cardiomyocyte, hepatocyte, and neurons, with all frozen state to pharmaceutical companies, not only in Japan but also in the United States and Europe. This cardiomyocyte is our first product in drug discovery. Our product is a frozen and single state. Once the frozen cell is sold, the cells can be reformed to make a thin layer. And if the thin layer is attached to the multi-electrode, we can detect these types of field potential electrocardiograms. The advantage of our cardiomyocyte is showing every time the high levels of potassium channels. When the acimizole or the cisapride is added on the cardiomyocyte, the QT prolongation is observed very clearly. The second cell type is hepatocyte. This cell have been developed by Dr. Kawabata by using adenovirus technologies. By using adenovirus technologies, the iPS cell can be accelerated into a mature hepatocyte. In conclusion, we have succeeded to develop the high purity hepatocyte, more than 90%. And the most important thing is activities. This figure shows CYP3A4 activities by adding the rifampicin. With an increase in rifampicin, you can see the CYP3A4 inductions. The fold induction is almost 7, which is comparable to cryopreserved primary hepatocyte. The last one is a disease model, which was jointly developed by Professor Nakatsuji. We have special homologous recombination technologies. By using this technology, any types of genes and disease genes can be integrated into iPS cells very easily. For example, presenilin-1 mutant is integrated into iPS cells and then differentiated into cholinergic neurons with high efficiency, more than 90%, to obtain the Alzheimer's disease models. This figure shows functional assays compared between the normal and abnormal disease models. The y-axis is production levels of Alzheimer's amyloid beta 42 productions. You can easily see the abnormal Alzheimer's disease models shows much higher amyloid beta 42 production than the normal one, which is consistent with Alzheimer's actual patients. That's all my presentation. Thank you.
Thank you very much, Dr. Yokoyama. Next speaker is Dr. Masaaki Toyoda, Chief Researcher, Research Development, Sumitomo Bakelite. And Dr. Toyoda, please.
Thank you, Chairman. I look forward to discussing our work concerning quantitative measurements of ES cell glycan profiles. Sumitomo Bakelite has developed innovative tools for regenerative medicine research. For example, prime surface stem full for stem cell culture and blood glycan array for glycan-related analysis. Here, we show glycan analysis of ES human cells using blood glyco. Generally, glycan analysis is challenging because glycan preparation is labor-intensive and time-consuming. As shown here, conventional methods require column chromatography and labeling, resulting in multi-step process that takes several days. For this reason, we developed simple and fast one-pot glycan purification and labeling kit blood glyco, resulting in preparation within 6 hours. Blood glyco kit is composed of hydrazide functional beads and is applicable to not only purified glycoproteins but also crude biological samples. Thus, this kit is applicable to comprehensive glycan analysis of cells and sera. Using blood glyco kit, we constructed workflow for comparative glycan analysis. First, glycans are prepared with blood glyco. The prepared glycans are analyzed by iCeMS, and the obtained data is extracted with profiling solution, and multivariate analysis is conducted using SIMCA-P. Glycan contributor to the discrimination of a comparative sample are defined by glycomotor tool. Using this workflow, we attempted to discover glycobiomarker candidates for quality control of human ES cells. Here, we compared glycan profile of undifferentiated and differentiated human ES cells called KHES1 cells. The analysis was conducted along previously discussed workflow. glycan contributors to the discrimination of the two states of ES cells are extracted. The figure on the left upper shows multivariate analysis score plot. Undifferentiated and differentiated groups are clearly divided in two clusters. The figure on the left lower shows glycan's contributors to the discrimination of the two groups. Glycan candidates are listed in order of highest contribution. Additionally, we conducted quantitative glycan analysis of glycan candidates. The figure on the right shows that LC peak area of the each glycan candidates displayed unique patterns in the two states. Moreover, Hymanus type glycans are the major contributor to the discrimination between two states. In conclusion, we developed a blood-like-based workflow for comparative glycan analysis. We found that Hymanus type glycans were determined to be glycobiomarker candidates for quality control of human ES cells by conducting the comparative glycan analysis of undifferentiated and differentiated ES cells. We are continuing to study the analysis of the discrimination of the early and late passage of the ES cells. Finally, ES cells were provided by Professor Dr. Nakatsuji and Associate Professor Dr. Suemori. This work was supported by NEDO. Thank you for your attention.
Thank you for Dr. Toyoda. Next speaker is Dr. Dr. Harumi Ginya, Technical Marketing Director, Genetein. Dr. Ginya, please.
Thank you for giving me such a good chance to speak about our technology and the product. Genetein is a daughter company of Precision System Science. This company, Precision System Science, is a robotic company. So we supply such OEM machine to Roche and Qiagen and Beckman. So we have a special patent about the BF separation of the magnet beads. We call this technology magnetization. So I will shortly explain this technology. So this is a scheme of the application of stem cells. So stem cell is cultured during the several passage. But usually many people use such technology. So microscopic observation, FACS scan, it based on the CD marker. Another is enzyme staining, cell staining. Another is a messenger RNA expression. But have you ever have such an experience that during the passage, stem cell changing are real, but you cannot detect anything that usual technologies. But we propose a new evaluation method. So I think that promising evaluation is epigenetic analysis. So especially DNA maturation, 5-methylcytosine and 5-methylcytosine is very promising. So unfortunately, I cannot show you the actual data because of patent issues, but we can get a very stable data. We can distinguish the stage of the cells. So I think that it become a good technology. And histone modification, we can do that. So this is a scheme of usual IP procedure. One is a chip, so another MEDIP and HMEDIP. So red is our machine can do that automatically. So if you use this system, you can remove the human difference, operation error, or operator's difference. You can get a very stable data. After that, you can use the high-speed DNA sequencer and microarray analysis. You can get a good data. So we can provide you a new automatic IP machine and protocol and reagent. So this is the actual machine. It's very compact. So XYZ dispensing machine, you don't have to make programming. So it is a GUI interface. What you only have to do is to put into some IP time and the temperature and the mixing speed. You can select these parameters only pushing the buttons. So I want to explain our magnetization. Magnet beads is a very hydrophobic, so it sticks into the chip's wall or tube walls. But this technology gathers the magnet beads very efficiently. It gives a very stable result. So this is the conclusion. DNA methylation is a very hopeful method. And automatic IP is a very good to get a very stable data. So last, you can get this machine from the two distributors. One is in US and Europe, you can get the Digen node. And Asia and Oceania, you can get ourselves Genetein. Thank you very much for the attention.
Thank you very much, Dr. Ginya. Next speaker is Dr. Masanari Kitagawa, Executive Officer, Vice President of Genetic Engineering Research Business Unit, General Manager, Dragon Genomics Center, Takara Bio. Dr. Kitagawa, please.
Oh, thank you, Chairman. I'm Kitagawa from Takara Bio. I appreciate everyone to give me a chance of introduce my company and products. So this is my company's profile. Our company is established as a result of a corporate separation through incorporation of a new company from Takara Shuzo. Takara Shuzo is a Japanese sake company, so the base of our technologies are standing at alcoholic fermentation. Then, December 2004, we were listed on the Mothers section of the Tokyo Stock Exchange. Since its beginnings as a biomedical business of Takara Shuzo Corporation, Takara Bio has developed biotechnology-related business with a focus on genetic engineering technologies. At present, we have other business segments. Cell and gene therapy business is developed. So now we have over 1,000 employees and about 20,000 million yen of net sales we have. So here I show our network. The sales network of the our business boosts global reach and encompassing North America and Europe and in Asia countries. So the business outside Japan become larger because of the Clontech, perhaps Clontech company as acquisition in 2005, and market growth in China is growing. So we have extensive lineup of Takara and Clontech branded products, and then we have a worldwide sales network. So the genetic engineering research business supports biotechnology research worldwide, not only basic research conducted at academic institutions, but also commercial entities working in fields such as drug discovery, research, and stem cell science. Since we began sales of the first domestically produced restriction enzyme in 1979, we are the first company introduced domestic restriction enzyme and PCR products in the Japanese market. And now we are we have offer 6,000 life science research reagents as catalog products in molecular biology or cell biology fields. Also, we have a contract research service capability using next-generation sequencing machines in Dragon Gem Center. We have continued to offer research reagents, scientific instruments, and contract research services that utilize the genetic engineering technologies. So, in such situation, we joined to the NEDO project. In this NEDO project, so in that, we play the roles development of profiling and evaluation reagents for quality control of human pluripotency stem cells. By the basis of the technology developed in the study of genetic engineering, those gene expression analysis and genomic analysis, including SNPs or LOH or CNV, and epigenetics and so on, then combine those analysis of the profiling ES cells or iPS cells, then we will establish a quality management approach for them. To do so, I believe there will be microarray or RT-qPCR and take advantage of the advanced technology such as next-generation sequencing. There is a proven experience and be able to demonstrate the power of us. So now we developed a new product as a part of this NEDO project. We have constructed that RT-qPCR primer set for analyzing the expression of genes associated with ES or iPS cells pluripotency. So this is based on the literature information on the International Stem Cell Initiative. The list of genes specifically revealed by the human embryonic stem cell was arranged. 88 stem cell-related revelation genes were selected. So we designed each primer in the design, not only the design of the primer, but also advanced verification of the performance to it by using real ES cells material from Kyoto University. So, sorry, yeah. Here I cannot have enough time, so for detail, I hope you're visiting my poster, number 93, and our booth. Thank you for attention.
Thank you for Dr. Kitagawa. All presenters are finished, and Question and answer will be can be obtained in our booth downstairs because limited time. So thank you for your attention.